Hello, fellow warriors. Here we are with news from the website prostatewarriors.com.
Absolutely, and there's a lot to cover today.
There really is. I mean, imagine finding out that an incredibly common, really inexpensive drug that we've used for decades to treat diabetes might actually be the missing shield you need to protect your body from the ravages of cancer therapy.
Yeah, or picture a scenario where scientists are, using gene editing to basically hack into the immune system's internal braking mechanism,
Right.
effectively turning a tumor's own fortress against itself.
It's wild. So welcome to this deep dive. Today, our mission is to navigate the rapidly shifting landscape of prostate cancer research.
And it is shifting fast.
It really is. And for you listening, we're going to strictly prioritize the science that is currently in human clinical trials first, so we can see what is actively being tested in patients right now before we transition to look at the most promising preclinical breakthroughs coming down the pipeline.
I think it's just a critical time to be unpacking this data because knowledge truly is a vital weapon in this fight.
Exactly.
When we look at the sheer variety of approaches on the table today, ranging from simple metabolic adjustments to CRISPR gene editing, all the way to advanced nanotechnology, it reveals just how relentlessly the scientific community is attacking this disease.
From every conceivable angle.
Right. It's a complex battle, but the level of innovation we're seeing, I think it provides a really profound sense of hopeful empowerment.
Yeah, I completely agree. So let's jump right into a treatment reality that many of you listening might already be intimately familiar with, and that's androgen deprivation therapy, or ADT. We have some fascinating news coming out of the PRIME Phase 3 trial.
Yeah, the PRIME Phase 3 trial is highly relevant because, ADT remains a foundational standard of care therapy for locally advanced and metastatic disease.
Right, the mechanism is pretty straightforward.
Exactly. It starves the cancer of the hormones, specifically androgens, that it needs to grow.
Mm.
But, and this is the big issue, that hormone suppression triggers a massive systemic shock.
Which is tough on the body.
Very. By drastically lowering testosterone, ADT essentially forces the body into an immediate metabolic remodeling.
Right.
And this is notoriously associated with triggering metabolic syndrome, severe insulin resistance, rapid weight gain, and a cascade of endocrine changes that really degrade long-term cardiovascular and metabolic health.
Right. So it stops the cancer from growing, but it's incredibly tough on the rest of the system.
Precisely.
So to address this, researchers in this trial wanted to see if they could mitigate those systemic side effects using an older, very common drug, which is Metformin.
Yeah, classic diabetes drug.
Right. So they enrolled 166 normoglycemic men, meaning, their blood sugar was completely normal, they didn't have diabetes, who were starting ADT. And they randomized these men to receive either metformin or a placebo alongside their hormone therapy.
And what makes this particular update so interesting is a correlative study that was derived from that trial. Researchers analyzed serum samples from 47 men taking the metformin and 32 on the placebo, and they tracked a comprehensive panel of biomarkers over a 12-month period.
Okay, and what did they find?
Well, the standout finding, the most consistent physiological effect they documented, involved a hormone called leptin.
Leptin. Okay, people usually associate leptin with appetite, right? Like telling your brain when you're full.
Yeah, that is its primary role in popular science, yes. Yeah. It's a satiety hormone produced by adipose tissue or fat cells. But in clinical biology, it's doing much more than just regulating appetite.
Oh, really?
Yeah, leptin is deeply intertwined with inflammatory signaling networks. So in the context of cancer, sustained high levels of leptin are linked to increased adiposity, worsened insulin resistance, and potentially much more aggressive prostate cancer biology.
Wow.
And in the placebo group of this trial, the men's leptin levels increased markedly over time, which was driven by the metabolic dysfunction caused by ADT. However, in the group taking metformin, that leptin rise was significantly blunted.
Okay, let's unpack this for a second. If ADT is like deploying heavy water hoses to put out a fire in your house, it suppresses the fire, but the water damage ruins the furniture and the floors. Metformin seems to act like a waterproof tarp, protecting the furniture from the worst of the collateral damage.
That's a great way to look at it.
Yeah.
If we push that analogy further, the tarp doesn't put out the fire itself, but it preserves the structural integrity of the house so you can actually keep living in it.
Right.
Metformin isn't acting as a direct cytotoxic agent killing cancer cells here. It's shielding your metabolic infrastructure. And, the evidence goes beyond just leptin.
What else did they see?
The metformin group also demonstrated favorable modifications in other vital markers like IL-6, which is a major cytokine that drives systemic inflammation. They also saw improvements in C-peptide and GLP-1.
Okay, I've heard of GLP-1 because of all those new weight loss drugs. But how do C-peptide and GLP-1 relate to this metabolic shield?
They're critical indicators of how your body handles insulin. GLP-1 helps stimulate insulin secretion when you eat, and C-peptide is a byproduct created when your pancreas produces insulin.
Okay, got it.
So by improving these markers, metformin is essentially helping the body maintain its insulin sensitivity. When you become insulin resistant on ADT, your pancreas pumps out more and more insulin to try and force your cells to absorb glucose.
And that's bad because...
Because that high insulin environment is highly inflammatory, it can actually promote cancer growth. By smoothing out that insulin signaling and reducing IL-6 inflammation, metformin is preventing the body from creating a microenvironment that the cancer could otherwise exploit.
So to be clear for you listening, we aren't saying metformin cures cancer, but this provides strong mechanistic evidence that adding it to hormone therapy reduces the metabolic burden, potentially really improving daily quality of life and long-term health.
Exactly. It makes the necessary foundational treatment much more tolerable for the patient.
That focus on mitigating side effects is so crucial. But if we want to talk about attacking the cancer directly, we have to look at how therapies are evolving to be more aggressive against the tumor itself.
Yes, the offensive strategies.
Right. There's a brand new drug currently in a phase 1/2 trial that's showing some dramatic results against advanced tumors, and it's doing so by targeting how cancer cells divide.
Right, so we are looking at AO252, which is a first-in-class TCC3 inhibitor.
KCC3, okay.
Yeah. To understand why this is a big deal, we kind of have to look at the mechanics of cell division. When a cell divides, it has to accurately pull its DNA apart into two new cells using these structures called spindle fibers.
Right, Biology 101.
Exactly. TC3 is a protein that stabilizes those spindle fibers. But in many cancers, TCC3 is heavily overexpressed, which is what allows the cancer cells to divide rapidly and recklessly.
So it's like putting cell division into overdrive.
Precisely. So by inhibiting TCC3, AO252 destabilizes the whole division process, causing the cancer cells to essentially scramble their own DNA and die.
What really caught my attention in the interim data from this phase 1/2 trial wasn't just the mechanism itself, but how a simple change in the dosing schedule completely transformed the results.
Yeah, the dosing was key.
They measured the clinical benefit rate, so the percentage of patients who saw their tumors shrink or stabilize. With once-daily dosing, the rate was 40%. But when they switched to twice-daily dosing in heavily pretreated patients, that rate jumped to 80%.
A massive leap.
Yeah. Why would splitting the dose make such a massive difference in efficacy?
It comes down to pharmacokinetics, really, and the continuous pressure required to stop cancer division. You have to remember, cancer cells do not take a break. They are constantly moving through the cell cycle.
Right.
If you give a drug once a day, the concentration in the bloodstream spikes and then gradually drops off. This is the drug's half-life.
Okay.
If the drug levels drop too low before the next daily dose, you create this window of opportunity where the TACI-C3 protein recovers and the cancer cells successfully divide.
Ah, I see.
By switching to a twice-daily regimen, the trial investigators ensured a sustained, continuous suppression of the target. They kept the pressure on around the clock, giving the tumor no chance to recover, which essentially doubled the efficacy.
And the duration of that efficacy is notable too, right? They're seeing response durations extending beyond 6 months, which in a salvage setting for patients who have exhausted other options, well, that usually only lasts 2 to 3 months.
The durability is really impressive, and it likely stems from the drug's secondary mechanism. AO252 doesn't just kill cells directly, it also triggers something called the CGAS sting pathway.
Okay, what is that?
Well, when the TACI-C3 inhibitor scrambles the cancer cell's DNA, fragments of that DNA spill out into the cell's cytoplasm where they just don't belong. The CGAS sting pathway acts as an internal biological burglar alarm that detects rogue DNA in the cytoplasm.
Oh, that's clever.
Right. Once activated, it alerts the body's immune system, essentially flagging the tumor for destruction by immune cells.
Wow. And I understand they're moving into expansion cohorts for prostate and ovarian cancers with about 30 to 40 patients, and they're rolling out a new formulation. They took the original tablet and turned it into a lipid-based soft gel.
Yes, the lipid system.
Is this similar to why some supplements require you to take them with a high-fat meal to actually absorb them? Like they're just giving the medicine a better vehicle to get into the bloodstream?
That is the exact underlying principle. The original tablet formulation hit a sort of dose-dependent bottleneck in the digestive tract.
Meaning it just couldn't get through.
Yeah. At higher doses, the body just couldn't absorb the compound efficiently or consistently into the bloodstream. By pre-dissolving the active pharmaceutical ingredient into a lipid or fat-based system within a soft gel, they completely bypass that absorption barrier.
Because our bodies love fat.
Exactly. The human digestive system is highly efficient at taking up lipids. The drug hitches a ride on those fats directly into circulation, ensuring reliable dose-proportional exposure. If the imperfect tablet yielded an 80% benefit rate, the optimized soft gel could theoretically drive even deeper responses.
That's incredible. Now, speaking of the immune system and that CGS sting alarm bell, there's another phase 1/2 trial making waves that takes a much more radical approach to immune activation.
Yes, indeed.
Instead of just sounding the alarm, this trial is actively reprogramming the immune cells themselves.
You're referring to CRG-150, which has just entered first-in-human testing for solid tumors, specifically targeting metastatic breast and prostate cancers.
Right.
It is a CRISPR-edited regulatory T-cell therapy, and to really appreciate how groundbreaking this is, we need to look at the immune system's natural checks and balances.
Let me make sure I have the basic immunology right here. Effector T cells are the aggressive soldiers, like the gas pedal, that go out and destroy threats. Regulatory T cells, or TREGs, act as the brakes. They suppress the immune response so it doesn't spiral out of control and attack healthy organs.
Spot on.
So if the goal is to fight cancer, why on earth are researchers using CRISPR to arm the brakes instead of the gas pedal?
It sounds counterintuitive, I know, but it's because solid tumors are incredibly adept at manipulating those exact brakes.
What do you mean?
A tumor isn't just a lump of rogue cells. It actively builds a highly suppressive microenvironment, a biological fortress, if you will. It secretes chemical signals that recruit your body's natural regulatory T cells to the tumor site.
Oh, wow.
Yeah. The tumor essentially hires your own immune brakes to stand guard, shutting down any effector T cells that try to infiltrate and attack.
So the tumor hijacks the body's peacekeeping force to protect itself.
Exactly that. So the researchers behind CRG-150 realized that simply infusing more effector T cells, pressing harder on the gas pedal, doesn't work if the tumor's brakes are too strong.
It's just spinning the tires.
Right. Instead, they extract a patient's TREGs and use CRISPR gene editing to reprogram their internal circuitry. The goal is to convert these suppressors into cells that actively support anti-tumor activity.
That's fascinating.
When these edited TREGs are reintroduced and migrate back to the tumor's fortress, instead of suppressing the immune response, they release pro-inflammatory signals that tear down the tumor's defenses from the inside out.
That sounds brilliant in theory, but permanently disabling the immune system's brakes must come with significant risks, right? I mean, if TREGs normally prevent autoimmune diseases, couldn't this therapy cause the body to attack its own healthy tissue?
That is the ultimate high-wire act of this therapy, and it is exactly why this phase 1/2 trial is monitoring safety just as rigorously as efficacy.
Right, they have to be careful.
Very. If the CRISPR edits cause the TREGs to lose all suppressive function systemically, you run a very real risk of severe autoimmune toxicity. The key will be seeing if the edited cells can maintain their anti-tumor activity specifically within the tumor microenvironment while leaving the rest of the body unharmed.
Okay, so we've explored how we can protect the body from treatment side effects, attack the tumor's cell division, and even hack the immune cells in the blood to fight back. But what happens when the cancer leaves the local environment and tries to spread?
That's the big question.
Right. That brings us to our next focus, shifting from active clinical trials to a major preclinical breakthrough out of Umeå University.
Yeah, the team at Umeå University has engineered a novel, fully human monoclonal antibody designed to block signaling through the TGF-beta receptor I.
And fully human is important, right?
It's crucial. Many older biologic drugs use proteins derived from mice or other animals, which can prompt the human immune system to eventually recognize the drug as a foreign invader and neutralize it.
Making it useless over time.
Right. A fully human antibody drastically lowers that immunogenicity risk, allowing for much longer-term therapy.
Okay, and the antibody targets TGF-beta, which the source material describes as having a very complex, almost contradictory role in prostate cancer. How does a single pathway both help and hurt the patient?
TGF-beta is arguably the ultimate double agent in cancer biology. In the early stages of a healthy prostate or even early-stage localized cancer, TGF-beta acts as a strict police officer.
Keeping things in check.
Yes. It regulates cell growth and actively induces cell death if things start growing out of control. It essentially suppresses tumor formation. However, as the cancer mutates and advances, the underlying biology completely flips.
Oh, man.
The cancer cells become resistant to the growth-suppressing effects, and instead, TGF-beta begins to drive a process called the epithelial to mesenchymal transition, or EMT.
What exactly happens during that transition?
Well, think of epithelial cells like bricks in a wall. They're meant to stay securely attached to their neighbors. Mesenchymal cells, on the other hand, are migratory. During EMT, TGF-beta effectively flips a genetic switch that causes the cancer cells to dissolve their structural bonds, gain mobility, and become highly invasive. It transforms them from stationary blocks into metastatic vehicles, while simultaneously suppressing the local immune system to help them escape.
So what does this all mean for you listening? Well, the principal cause of mortality in prostate cancer is metastatic disease, not localized tumor growth. A therapy that specifically targets the spread is basically the holy grail. So the Umeo antibody is specifically trying to stop that getaway process.
Yes, exactly. The antibody is engineered to target a specific pathologic cleavage event on the receptor itself. By locking down that receptor, it blocks the downstream signals that tell the cancer cells to undergo that transition and invade.
And it worked.
In their preclinical androgen-independent models, applying this antibody resulted in a striking inhibition of both primary tumor growth and metastatic spread.
The researchers noted that this could be a massive clinical tool if it translates successfully to humans. How do you see a well-tolerated antibody like this fitting into current treatment paradigms?
Its greatest potential really lies in combination therapy, because they observed no adverse effects on body weight or heart function in the animal models, which, by the way, has historically been a huge hurdle for TGF-beta inhibitors due to systemic toxicity. It could theoretically be paired safely with standard androgen receptor inhibitors.
Wow, that's promising.
Furthermore, it opens the door to combining it with targeted treatments like PSMA-targeted radioligand therapy, specifically 177-Lu PSMA 617.
Let's break down 177-Lu PSMA 617 for anyone unfamiliar. That is a therapy where they attach a radioactive isotope, lutetium-177, to a molecule that acts as a homing missile seeking out the PSMA protein on prostate cancer cells.
Right, delivering the payload.
It delivers a microdose of radiation directly to the tumor. So the strategy would be to use the antibody to stop the cancer from spreading, while the radioligand therapy acts as a targeted strike to destroy the existing tumor sites.
Exactly. You cut off the avenues of escape with the antibody, and you bombard the trapped cells with radiation. It is a highly coordinated strategy to prevent the disease from taking hold in new organs.
Which brings us to the most difficult scenario. The antibody we just discussed aims to prevent metastasis. But what if the cancer has already spread to the hardest, most impenetrable environment in the body, which is the bone?
Right, a huge challenge.
We have a revolutionary preclinical study published in the journal Small that attempts to solve this exact problem.
Bone metastases are the harsh reality for many men with advanced prostate cancer, and they present a really unique physiological nightmare for oncologists.
Why is that?
The microenvironment inside the bone actually protects the tumor from standard therapies. It's characterized by severe hypoxia, meaning very low oxygen levels, and it is heavily saturated with antioxidants, primarily one called glutathione.
Wait, we're always told antioxidants are good for us. Why are they a problem here?
It's paradoxical, right? But it's because many systemic therapies, like radiation and certain chemotherapies, kill cancer cells by inducing massive oxidative stress.
And antioxidants neutralize oxidative stress.
Exactly. So the cancer cells hiding in the bone use the local glutathione as a chemical shield, neutralizing the treatments meant to destroy them, while surviving perfectly well in that low oxygen bunker.
So to breach that bunker, researchers engineered a nanoparticle system designated PN20 at Rx. But getting a drug directly into the bone matrix seems incredibly difficult.
It requires a highly specialized delivery mechanism. The researchers incorporated a compound called alendronate into the nanoparticle structure.
Alendronate, like for osteoporosis?
Yes, alendronate is a bisphosphonate, typically used to treat osteoporosis because it naturally seeks out and binds tightly to the calcium in the bone matrix. By coating the nanoparticles with alendronate, it acts as a molecular homing beacon, guiding the particles out of the bloodstream and anchoring them directly to the metastatic sites in the bone.
Okay, so once the particles are anchored in the bone, how do they actually attack the tumor?
Through a process called sonodynamic therapy. Once the nanoparticles have accumulated at the tumor site, clinicians apply ultrasound waves from outside the body.
Just normal ultrasound.
Pretty much. Unlike radiation, ultrasound can penetrate deep tissues without causing damage. The acoustic energy from the ultrasound physically activates the sensitizers within the nanoparticles.
Oh, that's wild.
This activation triggers a dual action mechanism. It forcefully depletes the tumor's protective supply of glutathione, and it generates highly reactive oxygen species, completely reversing the hypoxic environment.
So this sounds like science fiction. We are essentially sending in a microscopic Trojan horse, the nanoparticle guided by alendronate, but the soldiers inside only jump out and attack when they hear a very specific acoustic signal from the ultrasound outside. That is wild.
The underlying mechanism there is brilliant because of its sheer precision. By stripping away the antioxidant shields and flooding the area with reactive oxygen, they create a sudden, overwhelming wave of oxidative stress that the cancer cells simply cannot survive, triggering multiple pathways of regulated cell death.
Amazing.
But the acoustic detonation goes one step further. The treatment also actively reduces the expression of a protein on the cancer cells called CD47.
What is the role of CD47?
CD47 is widely known in the field as a "don't eat me" signal. Cancer cells display this protein on their surface to trick the immune system's macrophages into ignoring them.
It's like camouflage.
Yes. By reducing CD47, the activated nanoparticles strip away the cancer's camouflage. As the tumor cells die from the oxidative stress, they release cellular debris that attracts immune cells. Without the CD47 signal telling the immune cells to back off, the macrophages sweep in and consume the remaining cancer cells.
Wow, a complete cleanup crew.
In their animal models, this entirely coordinated attack cleared tumors in the bone, prevented severe bone degradation, and even limited secondary spread to the lungs.
So if we step back and look at the whole picture, from using metformin to shield the metabolism, to optimizing doses for TESI3 inhibitors, hacking the immune brakes with CRISPR, preventing metastasis with antibodies, and using acoustic Trojan horses in the bone, what is the overarching theme here?
I think the most profound takeaway is that the era of searching for a single magic bullet to cure advanced cancer is over.
It's just not going to be one thing.
No. The future of oncology relies on orchestration. We are moving toward a reality where a patient's treatment regimen is algorithmically tailored. You might use metformin to protect the systemic metabolism, deploy a twice-daily lipid soft gel to aggressively halt tumor division, and utilize targeted ultrasound nanoparticles to clear out microscopic cells hiding in the bone.
Right.
The disease is no longer facing a single drug. It is facing a highly coordinated multi-domain siege.
A multi-domain siege. It really frames just how far the science has come and the incredible complexity of where it is going next. Notice how the future isn't about finding one single magic bullet to cure cancer. It's about orchestration. Imagine a future where a patient's regimen is algorithmically tailored, using metformin to protect their metabolism, a lipid soft gel to shrink the tumor, and targeted ultrasound nanoparticles to clean up the bone. Cancer isn't just facing a drug anymore, it's facing a multi-domain siege. Thank you for joining us on this deep dive into the latest research. I hope you found those aha moments, and remember that when it comes to navigating this landscape, knowledge is power. Stay strong and fight on.